Follow us on social media and please hit the subscribe button so you don't miss out on any updates from our ALS experts.
Okay, everyone, welcome again to your neighborhood. This is where we all gather, and this is what we call our clubhouse. And I'm making it a little bit light right now because it gets heavy quite often. And I just wanted to say to each of you, you each have a story, and it's very important that you share it. And I would just like to give you all a gift of my story. And so I've put up my audiobook on our website. And so if you go to everythingals.org and look at the videos, you'll see my 2 hours and 55 minutes of me reading my book, The Tale of an ALS Survivor. We've all got a story, and I used to be shy about sharing it, but no more. So I don't want each of you to be shy either. So tonight, when our open forum comes, don't be shy and share whatever you have. And with that, Lisa, I give it over to you.
Thank you, McFinn, and welcome everyone. I'm Lisa, for those who don't know me, and I am part of the Everything ALS team because I've lost my brother, so I continue to be his voice in the fight. So we want to thank you for joining us tonight for our ALS Talk series, where we provide our community access to the top experts around the globe. And we also connect the experts directly to our community to address the questions. So we also offer a safe place to meet and have open discussions with others that are going through similar journeys to yours. So we're honored to have our guest speaker tonight, Dr. Lyle Ostrow from Johns Hopkins University, who will be presenting on We Can Do Better, strategies to identify and validate effective ALS therapies and accelerate translation. So before Dr. Ostrow will talk, I would like to introduce Austin Edie Freeman. He is part of our Everything ALS team, and he is our patient advocate and industry advisor. So he's just going to share briefly how ALS has changed his life and his goal of leveraging his industry and patient experiences to transform patient care and end the horror of ALS. So Austin.
Thanks, Lisa. And I actually think it's a great introduction that McFinn gave today about everybody having their own story, because I think it's important for you all to understand who we are as an Everything ALS team. And who I am is an individual who has spent the vast majority of my career working in the pharmaceutical and biotech industries. And I also happen to be an ALS patient. I was diagnosed with ALS in September of 2019, 2 days before my 36th birthday. This was an incredibly devastating diagnosis, but I knew, given my career to that point, the fact that I'd always worked in industries that aided patients and people in need, I needed to do more, and I needed to continue to fight. So I was able to parlay my skill set into a very specific role at the pharmaceutical company I worked for. And then eventually, when I needed to step away, I was able to join the Everything ALS team. For me, it's so critical that we as pals have something to focus on even after diagnosis, because we can give back. We have a voice, and we are integral in the fight and the path to finding a cure for this terrible disease. I am so passionate about the work that's been done to date, the series that we've created through Everything ALS, our speech study, and what we've achieved through that. And I'm just very happy to be here today share a little bit about my background and experience, and welcome you to this session. And I know it's going to be a great one. So without further ado, I want to turn it back over to Lisa to introduce our.
Oh, thank you, Austin. Thanks for sharing your story and your efforts. And you know what? Everything ALS is so lucky to have you. So thank you for being a part of the team and being there. You're awesome. So I'd like to now present our speaker for tonight. So Dr. Lyle Ostrow is an assistant professor of neurology in the neuromuscular division at Johns Hopkins. His clinical practice and research are focused on ALS. He's the director of the JHU Neuromuscular Medicine Fellowship and JHU ALS Postmortem Core. He's also an assistant director of the Clinical Neuromuscular Pathology Lab, chair of the programmatic panel for the Department of Defense ALS Research Program. Dr. Ostrow has helped design and continues to direct many multi-centered collaborative studies using these resources focused on ALS and related disorders. So please join me in welcoming Dr. Ostrow. Thank you for joining us and sharing your insights and expertise on how we can do better.
Okay, thank you so much. Right, so I want to talk today about how we are truly working to try and do a better job, right? Get therapies that really, really work. And this is Jack Griffin. Jack Griffin's passed away. He was the chair of neurology at Hopkins when I first visited here as a med student, and then when I was a resident and then training. And he also was, at the time, he was the editor for Nature Clinical Practice Neurology. So this is an editorial he wrote back in 2006. And it says, research in neurological diseases lost in translation. The brain sciences are on a roll. Young scientists are choosing the neurosciences for their careers. Basic discoveries are accruing at a breathtaking rate. And yet, nevertheless, there is a palpable restlessness among disease advocacy groups, philanthropists, neurological investigators. And why? Because effective treatments simply are not emerging. We're learning a whole lot about these diseases, but somehow it's not translating into effective treatments. And, you know, this was written in 2006, but I think pretty much everyone on this call can relate, and it could have been written yesterday. And so what's wrong? And I'm going to get back to this editorial he wrote in a bit and some of his suggestions, but, you know, how can we do better? And why is it that there's this disease that we know so much about the basic science, and you know, there's so much research by all these super smart people around the globe, and yet it's not translating from laboratory experiments on, say, mice or cell cultures or genetic, you know, analyses into treatments for our patients. And so we have all this preclinical data, and it all sounds promising and exciting, and yet it fails to translate. And so we're going to talk some about what that means and why that might be. And, you know, some of the things that we ask a lot are these models that we're using, are these research models, are these cells in the lab, are things that we see in them really what's happening in people with ALS? Is it possible, are we choosing the right pals at the right times for these different treatments? What does it actually mean? Like, you hear about validating something all the time. Like, what does that word mean? What does validating mean? And how can we do it using laboratory resources actually from people with ALS? You know, part of this frustration, probably all this frustration, is that we've only got these couple treatments and they only slow the disease down a little bit. And we're desperate. And I like bolded and made big the we, and I want you to notice, like, we is not just people with ALS and caregivers with ALS. Like, you know, supposedly I'm an ALS expert, right? What does that mean if I can't treat my patients? If I don't have a therapy that works for my patients, what am I an expert in? You know, I'm an expert in telling somebody to maintain their weight and not fall and all the stuff that you guys hear about when you go to clinic, but I want to be an expert in how to treat this disease. And so I'm, you know, and everyone I know working on this disease truly is just as desperate as people with ALS and their families. And, you know, one of the things that's changed in the past 30 years since that definitely since that editorial was written even 15 years ago, is that we live in this interconnected world, right? So we find out about things going on all over the globe almost instantly. And it can be, you know, something great at the Olympics or something horrible in the Ukraine, but we're not doing a very good job of harnessing this interconnected world we live in to improve therapy development. We also have to, as we're learning from things like preprints and ways that information is getting disseminated very quickly, we have to be trained and understand how to tell what's meaningful and how meaningful it is and what the next step is in this data heavy world that we live in. So, I'm going to talk a little bit about kind of really what is ALS. And what I mean by that is, how do we approach this disease in the lab? Why do pharma companies decide that a certain thing is what they want to test as a treatment in ALS? Where do these treatments come from? Why do some things make it to trials and some don't? Why have so many trials failed? And this is really important, right? Because industry doesn't like investing money in this disease. I mean, every trial fails. They cost them millions and millions of dollars and it doesn't work. So how can we de-risk that investment? How can we help industry make investments in ALS clinical therapies that work? And everybody talks about biomarkers all the time now. Like, what does that really mean? And what kinds of biomarkers do we actually need? And what are the different kinds of biomarkers? And then importantly, how can we truly, collaborating, whatever, working? It sounds great, and everybody talks about it. But what is it? How can you actually do that in a boots on the ground way? How can we harness this interconnected world we live in to truly have a open science world as our lab kind of model? And I'll tell you, we are doing it and it's fun, and I'll explain in a bit. So, ALS is a clinical diagnosis, and this is part of the problem, right? We don't have a blood test for ALS. It's not like you can go and get your A1C measured when you have diabetes, or your blood pressure checked and say, oh, you've got high blood pressure. It's the diagnosis of ALS is based on your doctor telling you that you have ALS. And clinically, ALS, as you know, it's a disease of progressive weakness, right? And it can start anywhere in your body. It can start in your thumb. It can start in your breathing muscles. It can start in your foot. And then it spreads. And it's a fatal disease because eventually it involves muscles that control breathing and swallowing. And so it is a disease of this progressive weakness. It's something that we just recognize clinically. And so how do we recognize it clinically? I think Sarah said that she's in medical school and is about to enter the neurology part of her training. So, how does a neurologist diagnose ALS? Well, when a neurologist meets a patient, the first thing that we're doing, we're not trying to say, do you have ALS or Parkinson's disease or, multiple sclerosis or a brain tumor? The first thing we're trying to do is figure out where the problem is in your nervous system, right? And what's the tempo of the problem? And maybe it's more than one place. And so this cartoon here, this is my little cartoon of a brain and a spinal cord. And you've got nerves that are up in your brain, right? These are the upper motor neurons, and they come down the brain and through the spinal cord. They touch other nerves in the spinal cord and they go out to the muscles and they make the muscles move. And so when somebody has weakness, you can have weakness because of a problem anywhere along here, right? If you've known somebody who's had a stroke, they have a problem up in their brain and maybe they can't move some part of their body. Somebody who has a problem with their spinal cord or an injury or arthritis in their spinal cord might have weakness. Someone who has a problem with the nerve coming out of the spinal cord, that's neuropathy. That's like somebody with diabetes who has trouble moving their feet. And so when we look at this picture and when we look at where the problem is and try and figure out what we call localizing it, if you draw a line through this picture right here, and so one side of the picture is this nerve coming out of the spinal cord and connecting to the muscle, and the muscle. The other side is the brain and the spinal cord. This is the upper motor neuron. This is the lower motor neuron. And when we examine somebody, we see different things on exam. So if you have a problem on this side of the picture, you're stiff and spastic and you have jumpy reflexes. And maybe you have some trouble controlling your emotions. If you have a problem on this side of the picture, you see little twitchy muscles, right? The fasciculations and loss of reflexes and loss of muscle bulk. And ALS is a unique disease because you get problems in both of these things simultaneously in the same place, right? If you wanted to do that, if you wanted to point your finger to one place on this figure where you could get a problem on this side and on this side, where you'd point is to where they meet. And that's the anterior horn cell. That's the part of the nervous system that's affected in ALS. And the thing is, there's a lot of other stuff in your spinal cord and in your nerves and, for doing sensation and everything else that our bodies do. And somehow in ALS, it's just these motor pathways that are affected. And it's really the only disease that affects the upper and the lower motor neuron in the same segment of the body. So that's how we diagnose the disease. This is just fun pictures from Google kind of showing that same thing. So the motor pathway is called the cortical spinal pathway. because it goes from the cortex. The cortex is the outer covering of your brain. This is a picture of a brain here. It's looking this way, like the eyeballs are here. This is the back of the brain, the front of the brain. The part of your brain that controls your movement is this little strip right here. And it's got this thing called a homunculus. So different parts of it control your face, your arm, your leg. These upper motor neurons come down through the brain stem into the spinal cord. And then once they're in the spinal cord, they're in this thing called the lateral cortical spinal tract, because it goes from the cortex to the spinal cord. And then they touch other neurons and they go out to your muscles and they make them move. This is the lower motor neuron. This is the upper motor neuron. This is just a cut through the spinal cord of a different level, right? So a section of somebody's spinal cord. See all this stuff and all these words? Like there's a whole lot of stuff, right? And yet somehow none of this other stuff is affected, just the motor pathways in these two very specific areas. So the only way to truly confirm a diagnosis of ALS is after someone passes away and you look at their tissues under a microscope. And so this is sections of somebody's spinal cord, a photograph. This is a normal, and this is a person with ALS. And that part where it was traveling down, this upper motor neuron is pale, right? You can see that this is pale and this is not pale. And if you look at the part where the nerves touch, and that's where this cell body is of this lower motor neuron, that's what all these blue dots are. These are the spinal motor neuron cell bodies. And in the person with ALS, they're missing, right? There's a whole lot less of them. And if you look at somebody's muscle, this is what normal muscle looks like, sort of a cobblestone pathway. In an ALS muscle, you've got these really small angulated pointy fibers, because this is what happens to a muscle fiber when it loses its nerve. And so this is what we see under the microscope. This is what we see in a mouse with ALS. This is what we see in a laboratory assay or in patients' tissues. And as you guys know, when we talk about what causes ALS, 10% of ALS we call familial, right? 10% we think is due to a specific genetic cause that's inherited. 90% we call sporadic, and meaning that we don't actually know what the cause is. This is a paper from 2015. And so each of these things on this line represents a different gene that's been implicated in ALS, right? And the size of the ball gives you an idea of how important they are. So the most common mutation in ALS is C9orf. The second most common is SOD1. And as the years have gone by and we've learned different ways to analyze genetics, the number of genes that we've recognized has gone up. It's a little weird though, right? That there's so many different genes that can cause this very specific disease. This figure's from 2015. And this is an updated version from Dr. Harm. So I know you guys know, because I stole this off your website and because he gave a talk for Everything ALS. But he's added past 2015 on here. And so we're still finding genes that are implicated in ALS and lots of them. But I wanted to point something else out on this figure, and that's that. So this SOD1 was the first gene that was discovered, right? 1993. And look at that. There was a mouse model a year later. A year later, we had developed a mouse, which is still the most common mouse that we use in ALS research, where we put the human mutation of this gene in the mouse, and the mouse gets something that looks like ALS 1 year later. 1 year after that, Ryutech was approved. Now, Ryutech wasn't tested in this mouse, but you got to imagine, like if you're in this field in 1996, you're thinking, holy moly, we're going to have this ALS thing beat like right away. Look at this. We got our gene and we got our drug and we got our mouse model and we're good to go. And so, what's happened? Why hasn't things panned out the way we want them to be? And what's generated this? So, there's a lot of different things that have been implicated in ALS. So that stuff on the left there are just some examples of some of the different things that can go wrong in cells in your body that people have said might be involved in some types of ALS. And this picture in the middle, so the motor neurons are what die, but we now know that there's problems in a bunch of other different cells that are also in the nervous system or also help support the motor neurons. And we have laboratory experiments that you read about that, in mice, we take cells from our patients from blood or fibroblasts, and we turn those cells into brain cells and we can look at them. And, so where does this list come from? And before we talk about where it comes from, let me just say that just like it frustrates you guys, this makes my head explode, right? So this is crazy. How can all of these different mechanisms cause this very specific disease? And look at what the mechanisms are. And I mean, you don't have to be in medical school, like in high school, we like mitochondria, that's the powerhouse of the cell, right? I'm pretty sure all the cells in my body have mitochondria and really, really need them. A lot of these are very general cell mechanisms that are used by every cell in our body. So one of the things we truly don't understand still is how these different things, while they might cause ALS, what's the reason? What's the selective vulnerability of those motor neurons? And so where does that list come from? It comes from the way that we investigate things in a lab, right? So you have your disease and you have your normal and you compare them. And so sometimes and you look for something that's different, right? Sometimes it's really easy to tell, like an apple doesn't look like an orange. Sometimes it's harder to tell. Sometimes you might have two different varieties of apple and they don't look that different, but maybe they taste different or something is different about them if you do a chemical analysis. Sometimes you can't tell it all from looking at them and you have to use something like some weird imaging technique. These are both round fruit. Somebody put a peach and a pomegranate in an MRI scanner. I'm not sure why, but sometimes you need to use some sort of technology to tell the difference between your disease and your normal. And then what do? You take your disease and your normal and you say, okay, I see something that's different. And can I give a treatment to make the one that's the disease look like the normal? But the question is what you're seeing relevant to what's happening in a patient? So let's see. So this is an example. So this is just one study that, so these are mice, right? And these are that SOD mouse, that first mouse model that we've used since 1995. This is just one study that I was involved with. But so the idea is that the mouse on the left isn't treated and the mouse on the right is, right? And looks pretty good, man, right? Like you can say, oh, those mice are all running around a lot better than the ones on the left. But if you look really closely, maybe they are. But these are all litter mates. So on both sides, these are all litter mates and they're all this same breed of mice and they all have this same SOD1 mutation, but they're clearly not the same. I mean, it's almost like apples and oranges. You can see some of them are different colors, right? Some of them are big, some are small. These guys might be running around better than these guys, but like at least one of these guys over here looks like he's doing pretty good. And so, we've learned over the years that is this an effective treatment? I'm not sure. I hope it's going to be, but there's a whole lot of statistics and rigorous science that you have to do to figure out if it applies. And oh, by the way, this is one genetic mutation. And does this effect that we're seeing in this mouse right here, does this matter for somebody who doesn't have that genetic mutation? So this is my house. I live up in the country, like way off the grid in this old stone house that was built in the 1940s, a 2-foot thick stone. We moved here in 2013. The only reason we could afford this house was it had been vacant for 5 years and it was a disaster. Had 50 broken windows, mold in the basement, the heating didn't work, the plumbing was a mess, everything was broken. Our parents thought we were crazy for buying it, but it had a 50-foot pool and a woods and a pond and my little kid loved it. And so we bought it and we fixed it up and we spent a ton of time fixing it up and making it ours. And then we were all excited about it. And then that winter was a really cold winter in Baltimore. And what we learned was that this ancient stone house, if you're standing in the dining room and the wind blows outside, the wind blows inside. It's the draftiest, coldest house ever. And, if you didn't know what made a house cold and you just looked at our house and said, let's compare it to the neighbors. Let's see what's different about my house and the neighbor's house, the disease and the normal. The first thing you would notice is that we have a lot of ash in our fireplace because we burn a lot of fires, right? But if you didn't know what a fireplace was and you didn't know that it made heat and you said, oh, that's the problem, we got to get the ash out of the fireplace, that wouldn't help you. Now, interestingly enough, a lot of the things that we look at in basic cell biology are aggregates, right? Are stuff that's accumulating in places it doesn't belong, like TDP43 is one you might hear of a lot. TDP43 is a protein. That's what this top picture on the right is showing. It's normally in the middle of the cell, and in the disease, it goes out and it clumps. It's kind of like ash in a fireplace, except you got to be careful because is it doing something bad by being where it doesn't belong? Is it not doing something it's supposed to do by not being in the nucleus? Or is it just ash? And sometimes we know these things, sometimes we don't. Now, the other problem is, so that first year we burned a lot of fires. If you're sitting right near the fire, you're really warm. The rest of your house actually becomes much colder because it pulls in drafts, right? So we learned that really quickly. It's interesting, TDP43 is only found in a few cells in ALS, and you only need to find it in a few to make a diagnosis. But how does that explain what's going on, in the rest of a person? Now, if you understood how fires worked and you said, okay, that's, it's creating warmth and this is what we need to do. We need to burn more fires or turn up the heat. So we have an oil furnace in our house. And I think it's an okay furnace. If you compared it to my neighbor's furnace, I don't know if it's any different, but I know I burn a lot more oil. This is our oil tank outside and it's empty here. This is a picture I took in the middle of the night that first year when we were freezing because we ran out of oil. And again, if you compared my house and you said, okay, guess what? You need to burn less oil. That's your problem. That's the difference between you and the neighbor. It's not going to help heat my house. The fact is, a lot of those pathways that I showed you, a lot of those pathways that are implicated in ALS are kind of similar to a furnace, right? There are things involved in energy transport and, providing fuel. And so you always have to wonder, like, are these what's causing the problem, or are they your body trying to react to the problem? I guarantee my furnace breaks down a lot more than my neighbor's furnace. It doesn't mean it's a bad furnace, it's just working a lot harder. So, what I'm belaboring this point is that our body, our cells, are a bunch of interrelated pathways that try to compensate for something that's wrong. And we think that there's lots of different distinct causes of ALS. And, in most cases, the disease probably results from different things interacting with people in different patients, different people with ALS. And that's why a lot of efforts nowadays are focused on, can we figure out, are there subsets of patients? Are there people who have one thing wrong and not another, or one thing more wrong than another? Ideally, you'd like to address the underlying abnormality, like a specific gene mutation that you could perhaps correct. And importantly, we still don't understand how all these general mechanisms, all these different mutations, all these different cell types can produce this very specific picture. And oh, by the way, it's even worse than this, right? Because many of these same mechanisms are implicated in other diseases. Alzheimer's disease, frontotemporal dementia, but even things like prostate cancer or liver disease, things completely unrelated. So how does that work? So in my house, the genetic mutation is that we've got these thin single pane windows, right? These steel sash 1940s windows. I can't do anything about them because they're in 2-foot thick stone. Gene therapy is hard, right? And I can't fix that part. And so that's not really an option for me. Gene therapy is getting easier, by the way, as you guys well know. This is my father-in-law. He's passed away now, but this is so they used to come visit us in the holidays, and they were always complaining it was too cold and drafty and turning the heat up, and we were spending hundreds of dollars on oil. And so one year for Christmas, we bought him this fantastic footy pajamas off some infomercial. And this is not affecting my windows. This is not fixing the genetic problem, right? My steel sash windows. But guess what? This is a good treatment. He doesn't feel cold. It doesn't bother him. He's completely happy and smiling. It had this, side effect like some medicines do, that it made him look hilarious. So we all liked it too. But this is what tirasemtiv or reldesemtiv, which is the newer tirasemtiv and currently in trials, is doing, right? So this is not treating the cause of the problem, but it is helping the remaining myofibers, the remaining muscle, be more efficient at contraction. And, this is a good therapeutic strategy. It doesn't necessarily have to affect the actual problem. So why have so many trials failed? And I'm going to use the example of ceftriaxone, which was a few years ago now. So one of the big ideas in ALS is this idea about what's called glutamate excitotoxicity. So glutamate is this thing that excites your nerves. And in ALS, it looks like there's too much of it. And it looks like there's too much of it because the things in your brain that usually get rid of the excess aren't working right. And so there was a lab assay in cells. And the lab assay was an assay for things that would help increase those what are called transporters to get rid of the excess glutamate. And they screened all these drugs. And the thing that fell out was ceftriaxone. And this picture on the right, so this is a study in those same SOD mice. And you guys see this kind of stuff a lot, I'm sure. But so this is looking at survival, how many mice are surviving in a colony. And this is looking at how long they're surviving. And the ones that got the drug, lived longer than the ones who didn't get the drug, who got the control. And so this got taken to patients, and there were 2 phase 2 trials, and they both looked like they had an effect, and there was a lot of excitement. And then there was a big phase 3 trial, and the phase 3 trial failed miserably. Didn't work, got stopped early, didn't work. So why did it fail, right? Did it fail because it was a dumb idea? Did it fail because, it just was not a right idea, or maybe it only would work in a subset of patients, or maybe the drug didn't get where it needed to go? Or maybe, so this is that transporter it's supposed to be turning up to help get rid of that excess glutamate. Maybe it didn't do that. Maybe it got there, but just didn't do it enough. And so if we'd had a way during that trial to track the levels of that thing it was supposed to be doing, maybe we wouldn't know that answer. As it is today, we don't know. We don't know. Is it a bad idea? We still don't know. We don't have a clue. And, this is where biomarkers come in. And this, again, is not a new concept. And everybody's been talking about biomarkers in ALS for a while. This was a meeting put on by the ALS Association back in 2014, where we all got together to talk about, what can we do? What's a biomarker? And these are quotes from this press release. So any measurement that tracks the disease process, we were kind of simplistic back then. We've learned more about how to think about these things. And the big idea was that we needed to find a way so that pharmaceutical companies were willing to invest in ALS, that these biomarkers were a big part of de-risking. Tell whether a drug has reached the target and done what it's supposed to do. And I'll tell you, in this age of big data that we have right now, where everybody's measuring everything from everything in patient samples, you have to be careful, right? Because, so this is a website that I encourage you to go play on. It's called Spurious Correlations, tylerregion.com. Takes public data sets and mines them for correlations. So look at that. The divorce rate in Maine correlates with the per capita consumption of margarine. I don't think that that's causative. The US consumption of cheese correlates with the number of people who died by becoming tangled in their bed sheets. From the USDA and the CDC over a decade, that's a better correlation than a lot of ALS studies. So it's just, you have to be careful. You have to, when we're measuring so many different things in so many patients. Imaging studies can be biomarkers, right? So you can look for changes in somebody's MRI. This is expensive, and you've got to do it the same way at different sites. And MRIs take a long time to schedule. And like if I bump my elbow and I want to get an MRI, it might take 3 months, and maybe my elbow doesn't hurt anymore. And so that's a problem for clinical trials. PET scanning. So PET scanning is where you take a radioactive substance that labels a specific thing and you inject it into someone. And it can be great because you can label the target you're trying to hit with a trial, and you can say, oh, does my drug get there and affect it? But again, it's expensive, and it has to be done the same at different places. A few years ago, we ran a PET study trial at Hopkins for a particular receptor. It doesn't matter what it was, but the idea is that if you do these PET scans and you look at a healthy subject or an ALS subject, and you can compare where it goes, and you can see if there's differences, and you can see if medicine changes the differences. We learned a lot about that study. This was several years ago now, when people weren't so savvy with this stuff. And one of the things we learned is that people with ALS didn't want to enroll in a biomarker study because they were worried that it wasn't a treatment and it would make it so that they couldn't enroll in a clinical trial of a treatment. And I understood that. It wasn't the case, but we needed to educate them. We learned that, as ALS progresses and somebody can't lie flat because they have breathing trouble, that's an issue if you're doing a study that requires somebody to lie flat in a scanner and still. It was a challenge to learn how to coordinate these kinds of things. So this is the kind of information we learned about how to do these particular studies. We also learned that, at least I personally learned, that 90% of my ALS patients were smoking marijuana, which is fine by me, but it meant that they failed all their tox screens for the PET center, and we had to deal with that. So there actually are several different kinds of biomarkers, and it's really important that we understand this. And these figures are from Michael Benatar, and I know he's spoken to you guys too. So the simplest idea of a biomarker is something that is elevated in a disease and not elevated in somebody who doesn't have the disease, right? And this is a diagnostic biomarker. And I said we don't have this in ALS, but to be totally honest with you, we're not terrible at recognizing ALS. We're pretty good at it. We'd like to do it earlier. We'd like to have a diagnostic biomarker, but this is not necessarily the reason that the trials are not being more successful. So what would you call a biomarker like this? So so you have a treatment that, let's say the treatment is a genetic therapy for people who have a specific SOD mutation. So if that's the case, you don't want to enroll somebody in that trial that doesn't have that mutation, right? And so the gene test for that mutation is a kind of a biomarker, and it's called a predictive biomarker because it predicts who's going to respond to that therapy. Now, understand something. So a predictive biomarker doesn't have to be for a gene. Like, let's say you have a therapy that is something that targets inflammation in the brain, or mitochondrial, trafficking in your nerve cells, or whatever it might be. You'd like to have a lab test where you can figure out which people with ALS have a lot of that thing, right? You'd like to enroll the patients who are most likely to respond to a particular therapy that's targeting a specific pathway. So this is a different kind of biomarker again. So something you measure one time, and it's either low or high. And maybe if it's high, you're likely to survive longer. Or maybe if it's high, you might have a higher function, like your strength might stay longer. And so a one-time measurement that tells you if somebody might have, let's say, fast or slow progressing disease. And so this is what we call a prognostic biomarker. And this is important for matching cohorts in a study, right? You want to know that you're enrolling patients that look like each other. That if you have patients who progress slowly or quickly, that they you know who's who, and you're enrolling matched patients. So this one here, so each of these lines is a patient, and And so in this particular case, you have something that you're measuring in a patient, let's say their blood or their spinal fluid, and it stays the same, or maybe it's slowly going up as the disease progresses, but then you give a therapy and the one that was the same goes down. And it's different in different patients. Some are high, some are low, and some go down more or less. Maybe if it was going up, maybe it stops going up. And so the idea here is it's something that lets us detect when a therapy did something that we wanted to do. And so this is a pharmacodynamic biomarker. And you guys hear a lot about, for example, neurofilament, right? Is neurofilament a pharmacodynamic biomarker? It is in some diseases, maybe in ALS too, meaning that we can see a change in it with the therapy. But what we'd really like is pharmacodynamic markers that are specific to a drug, right? If your drug targets a specific process, you'd like to know, do you see a change in that process when you give the drug? So why are patient-derived samples important? Patient bloods and patient tissues. And the point is that these models that we have that are based on various, mutations or flavors of ALS, no model is ever going to represent the findings of every person with ALS. I mentioned TDP43, right? The most common thing that people talk about in microscope, looking through a microscope at ALS tissues. And I mentioned the SOD mouse, the most common mouse that we use, right? Well, guess what? SOD mice and SOD patients are like the one exception where there's no TDP43. So, the mouse model that we use most frequently in the lab, in fact, doesn't have the pathology that we think is most common in ALS patients. So it's important to know, are changes that we see in the lab important, and are they happening in our patients? And this is the thing. So if I take an apple and I grind it up and measure something in it, that's applesauce, right? But tissues in our body are more like apple pie. There's all different kinds of cells in them. And in fact, the neurons, as the disease progresses, are going away. They're dying. I showed you a picture of that. And so the amount of apple in the pie is decreasing. And if I measure something in the blood or the spinal fluid, I'm not actually measuring the pie, right? I'm measuring the stuff that's dribbling off onto the plate and going away in the blood vessels. And so it's really important that we're able to know, are the things that we see in lab that are changing, can we measure something that correlates with that while somebody's alive? And so that's where this, this need for this very rigorous standardized data comes linked to samples. And I know lots of you on this call are involved in things like, whether it's the CREATE Consortium or, ALS TDI or Answer ALS, these really, really amazing, highly curated studies or the CDC that are collecting samples and data longitudinally. And one thing that there remains very much of an unmet need for in research is the ability to match those tissues and those data and those samples to autopsy tissues afterlife. And also just to have enough of those samples for researchers to use and to understand what's changing to figure this stuff out. So back to that essay that Jack wrote. So, his essay concluded with a list of strategies. And again, this is in 2006, right? And he's talking about, setting flexible funds and having unfettered exchange of ideas and data and new intellectual property approaches. This all sounds great, right? This is the stuff everybody talks about in all these meetings, but it's hard to do, right? It's hard to operationalize this. Like, what does it look like? How do you actually do it? And it's frustrating that this was in 2006 and it still feels that way. And what does it really take to get all of us in the sandbox together and bringing our own expertise and our own tools to, build the best sandcastle? And so this was the inspiration for, in 2013, when I founded and directed this multi-centered post-mortem tissue effort to collect and analyze these tissues. And so we were doing this with standardized procedures across a bunch of different academic institutions. And we had centralized, genomic studies at the New York Genome Center, the ALS Consortium, and data management by the Neurobank team. And, our goal was to provide the tools that researchers need and to do it in a way that respected the donation. Because guess what? If somebody who I'm treating, my patient, donates their tissues to my research, well, that's the most amazing donation there is. And we need to make sure it's used responsibly and appropriately and as much as possible. And so we sent out all these surveys and we figured out, how can we do this dissection? How can we do the quality control? How can we measure everything that we can to make sure that each autopsy is used as much as possible? We got centralized genetics done in a highly curated way. We made all this data, all these samples freely and openly available to whomever wanted to use them. We didn't ask for anything, no authorship, ownership, intellectual property, nothing. This was all just openly available to whomever wanted to use it. We collected tissues in different ways. We had a barcoded system. This evolved as it went because the needs of researchers evolved, the way they had to be processed, what was important. We had pathology that was standardized. And then this is where it really got interesting, because what we realized was, so we had this request form, right, and a committee, and we're going to review these requests, but who are we to review an idea when nothing is working, right? So what we decided to do, instead of having an evaluation of, say, whether somebody's science is a good idea, was to evaluate for feasibility. Are the experiments well designed? Can they do what they say they're going to do? Have they justified the amounts and what they need? And so we actually abolished the committee, and it just became a rolling review by me based on these criteria. And every week I get several requests, and I still do all the time. And we can do this in a matter of weeks rather than months or years or whatever it normally takes. We have standardized MTAs. We have no intellectual property requirements. And again, we are reviewing based on feasibility. And importantly, we also spend a lot of time talking to researchers about their experiments and figuring out, can we do a first experiment that's going to help inform the later ones? And can we decide early on in the science, what's going to be the biomarker in a clinical trial? And can we look at that? Can we say, take a look at, measuring something in parallel to whatever it is that they're measuring that's going to help eventually with that clinical trial? So that first study I showed you, what was my role in it is that, we were using muscle samples from these patients that were in this core. And so this thing that MG53 causes in the mouse, in the SOD mouse, these weird bubbly, messy things, right, that aren't in the wild type, and using human, tissues from people with ALS, we saw the same stuff. And we saw the same stuff, not just in SOD mice, but in sporadic mice, right? Sporadic people who didn't have a genetic mutation or had a C9 mutation. And so this is the way that we can look and see, is what we're seeing in an animal model the same as what's going on in people with ALS. And so based on that work, this woman who was the one who ran these studies, so Jinsung Zhu, who, by the way, was not an ALS researcher. We helped her. We had weekly meetings with her, teaching her in essence about ALS at that time, but she now has a grant and is carrying this forward as a potential therapy. And I hope you hear more about it soon, actually. We spend a lot of time talking to researchers about designing their experiments, about, as I said, that, if you're doing something in a cell line, that's apples and tissue's apple pie. And so how can we make those two things look like each other? What does it really mean to validate? What is it that they're trying to do? What's the go, no go decision? How does the company decide what they're going to do next? We have learned a lot about how to use different parts of the human nervous system for different kinds of experiments in ALS. We've learned about what to compare. So it turns out it doesn't make sense to compare, in most cases, an ALS, a person with ALS, to somebody who doesn't have any disease, right? It makes a lot more sense to compare maybe a part of the brain of that person to a part of the brain that's not involved in the disease, because a lot of these pathways affect lots of different disorders. And we've learned a lot about how to collect the data and how to harmonize the data so that all these big studies doing things are collecting so that it means the same things, right? And so I'm going to skip through this, but we design experiments with everyone who comes to us, and we try to screw them up. We try to figure out how to make their experiments fail. These are some examples of projects that have been using our tissues. Many thousands of samples, 120 different labs across the world, over 700 terabytes of genomic data. As of this year, we at Hopkins have separated from the multi-centered effort and formed a fully academic effort. We still work, everybody collaborates with each other. Doesn't actually matter because everything's given away openly and freely. But this is a collaboration with the CDC National Registry. And I hope to be able to tell you very shortly about it's going to make us able to, for example, collect autopsies from people who are everywhere and not just at the sites that are involved in these studies. These are some of the labs that have used our tissue samples and slides. I get 4 or 5 new requests almost every week. These are some of the places that have used the raw genetic data. Again, it's all provided openly and freely. As an example of how open science works like this, so this was one of the first papers that was published with these core resources. This was work by Avi Nath at NIH on something called endogenous retroviruses. These are viruses that were infected your ancestors thousands of years ago and left junk in your DNA. And in some diseases, including ALS, this stuff gets turned on and causes a problem. And so he found this stuff in the tissues from us, and then he made a mouse and based on this stuff, and the mouse looked like it had ALS. And this paper gets published. And almost immediately after it got published, every big pharma company that has HIV drugs were like, whoa, that thing's a retrovirus. It's like HIV. We could use our HIV drugs to treat ALS. And they all contacted me and they said, give us all your samples because we want to study them ourselves and do our own genetic analysis. And we were like, yikes, that would be horrible and it would not be responsible. And we learned that in order to detect these specific weird things, you had to do the genetics in a like more expensive and more difficult way. And so instead, Hamali Fatnani and I, so she's the director of the ALS consortium at the Neurogenome Center. We just called the ALS Association and that was helping to fund this. And we said, hey, we don't want to do it this way. Would you be willing to fund us to basically do this other kind of analysis and then make it broadly available? And they said, of course, sure. So that's what we did. As the science progressed, within a couple of years, in fact, these endogenous retroviruses are involved in all sorts of diseases, but by 2017, there was a session at their biannual meeting specifically on ALS. And they asked me to chair it, even though I knew nothing about the science, but just because all these people were working with us. And this is Avi Nath, by the way. At that meeting, we met this woman, Molly Hamel. So Molly Hamel at Cold Spring Harbor has a lab. She is like the world expert in doing this super amazing analysis of these weird things in RNA. And so we said, hey, Dr. Hamel, we will give you all this data for free if you do your fancy analysis on it. And she said, wow, really? Cool. And so she then gave us her analysis back. And she asked me something weird when we met. She said, is it okay if I find things besides these retro elements? Because we often find other things. I didn't understand what she meant. And what she meant was normally if somebody gives you a resource for a particular purpose, like they get mad if you do something else with it. And I was like, no, this is free, open. We just need to do this. And so indeed, what she found by analyzing our data was retro elements, but also that we could divide all of these different tissues into different clusters. And the different people with ALS, when we looked in their tissues, had different things wrong with them, different pathways. And so now the question is, can we find things in somebody's life, in their blood, in their muscle, in their spinal fluid that might correspond? Because if I have a target, if I have a drug that targets a particular pathway, I want to make sure that I'm enrolling the right patients. So this was published as a preprint, right? You guys know about these things now. This is this new thing. And the day that it was published as a preprint, this was a request that I got from a pharma company on the same day. And so this is how you accelerate science, right? You get the data out there, people can use it immediately. They wanted to do this because they wanted to see if they could figure out who the subgroup was that was going to respond to their therapy. As this progressed, there's now at least 5 ongoing projects, 10 institutions, 3 clinical trials that have been directly inspired by this work on these clusters. Dr. Hamel, for this work, got an award from the Chan Zuckerberg Institute. And because of that, they invited me to come talk about this way that we're doing this open science at their kickoff meeting a few years ago. And so now we're working with several other of their researchers in their labs. And NIA and NINDS last year asked us to come and talk to them about this stuff and how can we work together to work on it. And so this is what it looks like when things are shared openly and freely. This was, so March 2019, this is a meeting of the CREATE Consortium in Florida, which is another one of these wonderful multi-centered efforts, collecting samples, doing all kinds of analysis. Why was I there? I was there because we gave them our data. They did an analysis. They were using it to build a platform for ALS. And it was actually, I was at this meeting when that preprint came out. And I told you that I got that request immediately. The pharma company that sent me that request was at this meeting. They were sitting 2 rows behind me. And I walked over to them at the break. I'm like, dude, what's up? You can just talk to me. And they're like, yeah, we've all got the paper. Everybody's talking about it, right? So this is what science is supposed to look like. These are a few just excerpts from the kind of data requests that we get. Can we use your data to figure out what's different, clinical heterogeneity between different patients, which drugs are going to work in which people? Can we find biomarkers that can be tracked for rescue with different treatments in our patients? So the key is we need to encourage biomarker development, biomarker translation, right, in clinical trials. As you guys know, we actually have a lot of candidate biomarkers in ALS, lots of different things that we measure. What does it mean to actually make them ready for a clinical trial? And so at this meeting, the other reason I was there was that Michael Benatar and I designed this study for Neurofilament where we said, look, we got all this data, but we need to know, like, how does it behave in different laboratories, in different companies? And so we found every clinical research organization that runs every commercial test that measures these things. And we sent the CREATE Consortium samples to them. And we did it all blinded. And we compared them all because we needed to know which ones work differently, which ones work in different hands, different labs, right? This is what you have to do if you're going to use it in a clinical trial. We made everything freely available. So the data is all on Data Dryad. This was also published as a preprint, October 10th, 2019. Less than a year later, I was participating in this webinar about preprints, right? Because the COVID made everybody understand preprints, because this has become a much more important thing. I wasn't there because I know anything about COVID. I was there as representing as a researcher who's used preprints and these sorts of methods to help with open science. But the interesting thing was, so this is less than a year later, and this was just an excerpt from the slides I presented. And already it's those results that we put out as a preprint are being used in these big reviews. They already were incorporated into 25 different trials, and it's much more nowadays. So back to Dr. Hamel. Now we're talking with them because we want to analyze muscle, because we have muscle from our same people who died from ALS, and we looked in their brains, because guess what? Maybe we can find something in muscle, for example, that correlates to what we saw in the brain. And maybe that could be used in life to help figure out who to give what treatments to. And I'm going to skip through this, but muscle biomarkers are something that there's a fair amount of history about, and you can time them for different treatments or different, biofluids. It's not generally part of the diagnostic workup for ALS, but there are clinical trials that have used it. So right now, the Helix Smith trial, we did muscle biopsies before and after patients got treated to see if they got changed. I do muscle biopsies for clinical diseases at Hopkins, and we take muscle out of the patients. Not necessarily ALS, although we can for ALS, but this is for a different disease, inclusion body myositis. And we take that muscle and we transplant it into the legs of mice. And it grows in the mouse's leg. So this is a picture of a mouse's leg. This is human muscle growing in a mouse's muscle. It gets innervated by mouse nerves and mouse blood vessels. And you can put normal muscle in one leg and disease muscle in the other leg. And you can give the mice drugs, and you can look at things that are secreted into the blood. And you can use this to look for biomarkers and to test treatments. And so this particular disease, IBM, this is a paper that just came out a few weeks ago. And yeah, this isn't ALS, it's IBM. But guess what? This paper is using these xenografts to look at TDP43, right? We've heard of that. And so, these are things that can be sampled during a patient's life. You can do it from diseased mice, you can do it from diseased humans to help with this biomarker development. As another example of something super fun, so one of the labs we're working with right now to get them tissues is Amidus. Amidus has this TDP43 retinal marker, and to help them develop it, we were helping them get eye tissue from people who had died from ALS. And what's interesting is that this woman who had that other thing I told you about, the MG53, so she's now doing work on muscles, eye muscles, right? Because eye muscles are spared in ALS. And we went back to Amidus recently and said, hey, can you guys give us back the muscle part of those eyes we gave you? Because you don't need it. And she could really use it. And they said, yeah, sure. And so why am I sort of talking about all this stuff? Because what's the pie in the sky? Like, the world is our lab right now. Okay, this is how it works. And I've just given you a couple examples, and there's more all the time. So earlier this week, I had a call with 2 different groups who are developing voice apps for tracking patients with ALS. Sound familiar, right? I think, might be something that Everything ALS is interested in too. And over the years, what we've learned is it's really easy to get people into the same room. Nobody ever says no, and to share expertise and to do it via Zoom and literally to have these virtual lab meetings that we've been doing now since 2013. It's been amusing to watch everyone else learn how to use Zoom and really have this world as our lab model. And so it works. So how can we think about biomarkers even earlier though? So I teach the medical students, I teach the graduate students about ALS. I teach them all the basic science that about all the different experiments, but then this is the exam question I give them every year, and they all know it's coming. I never change it, right? I don't ask them about the different mechanisms. I say, choose whatever you want or propose something new, and then tell me how you're going to design an experiment to test it, and tell me what your biomarker is going to be and how you're going to figure out if that biomarker works, right? Because we need the next generation of scientists thinking about this stuff, understanding that this is what's necessary to translate. One of the greatest funds I have in ALS and most invigorating things is spending time with people and caregivers with ALS at the Curleys, at the Clinical Research Learning Institutes. And, it's important that we talk about, for example, the importance of participating in trials that have biosample collection, right? And that these trials are critical to help us figure out how to do better with developing therapies, and the importance of autopsies too. And that, the thing that everybody wants is to have the blood and the, data collected during life, and then have the tissues from those same people. And look, that's the ultimate donation, and we get it, but it really, really matters. And so this sort of outreach is really important. We're also, well, I'll get to that in a sec. So the other hat that I wear in this community is that I'm the chair for the Department of Defense ALS Research Program, right? And so this is the website, and I encourage you to go take a look at it. And for those of you who are not familiar with it, we're presently the largest dedicated funder for ALS therapeutic development. So this is part of the DoD, part of what's called the Cdmrp, Congressionally Directed Medical Research Program. And this actually came out of a grassroots effort, right? So originally it was for breast cancer, and a bunch of advocates got together and convinced Congress to fund this program. And from the very beginning, this program had a 2-tier process, scientific programmatic reviews, and fully integrated what the DoD calls consumers. Consumers are pals and cows, right? So every aspect of every Cdmrp and every disease involves people with ALS, caregivers with ALS. Over the years, other diseases got added, including ALS. The hallmark of all of them is the involvement of consumers in every stage. So the ALS one, up through 2019, had 2 mechanisms. And the one mechanism was to find innovative new therapies. The other mechanism was to do the unsexy stuff, to take therapies that were being developed in the lab and get them to what's called an IND, to get them to a clinical trial, right? To do the tox and the good manufacturing and all the processes that were needed. And those were the only 2 mechanisms that when I joined actually this program in 2013. And the reason we chose those 2 was that with the budget that we had, that was where we felt we could make the biggest impact, because we had NIH funding this stuff, and it was a part of this pipeline that was underfunded. And so I joined here, and this program didn't really work so great back then, because we would get 100 applications and we could fund 2 or 3, and it really wasn't very successful. More recently, the budget has gone up tremendously thanks to the efforts of advocates like you. And when we learned that the budget was going to go up, so let me show you. So this is all the different diseases, right? And so some of them are huge, like prostate cancer, it's peer-reviewed medical, whereas breast cancer, some of them are much larger. These are the ones that have been successful in funding large clinical trials, getting things from the lab to the clinic, these very large programs. One of the nice things about that is that we don't have to ever reinvent the wheel. Whenever we're refining or adding mechanisms, we've got all of the experiences of all of these other programs. So when we learned that the budget was likely going to be increasing, we didn't want to be like 7 people in a room figuring out what to do. And so we did what we always do, which is crowdsource the wisdom of the entire community, right? So I wrote a survey. Some of you may have participated in it. Went out to everyone. Went out to researchers, clinicians, patients. We were asking, how much of an understanding is there of the mechanisms? How can we improve them? What should we do? We talked about biomarkers, data sharing, all these kinds of things. And based on that year that our budget increased to 20 million, we added a new award. We added this clinical development award. 20 million was not enough for us to realistically fund all this innovative therapy discovery and big clinical trials, but it was enough for us to fund adding on an aim to existing trials to collect samples or do biomarker analysis. We could fund small trials that were looking to optimize components of current ALS care, right? Like what's the best way to use a ventilator, or what's the setting on your CPAP machine, things that we don't really know. And so this was this first year that we added this. And there was a tremendous response, by the way. But then the next year, we found out that the budget was going to be increased again. And by now, we had this huge portfolio of innovative things that were about to be ready for clinical trials, right? Because that's what we had prioritized. And these were the kind of things that were well situated to maybe move into early clinical trials and then beyond. And so we said, okay, we now have enough funds based on the experience of these other programs, based on surveying the community, that we added a biomarker-driven therapeutic clinical trial award. This was the first year that we did it. We also further refined the awards that we had, and we do this every year to make them. So for example, you get more money if you're including a biomarker, and data sharing is now a requirement. Now, this year again, so once a year, we do what's called programmatic review. And this year we did that again. And part of it this year, we wanted to know, again, what's the clinical trial landscape look like? How can we most have an impact? What kind of things should we be funding? What's the funding landscape look like? How has COVID affected it? We now have added industry representation to the panel. And so what can we do for industry to help de-risk, but also how can we improve outreach so that industry labs are aware of these mechanisms and that they can apply for them? And so, Nadia Sethi from I'm ALS and now ALS TDI, was one of the people who led the development of this fantastic clinical trial ALS signal database, right? And so we gave Nadia a specific task. We asked her to survey the landscape, and we had a specific question. We wanted to know what percentage of trials are using biomarkers, what kind of biomarkers are they using, what sort of phase are they in, and we also, if you go on the ALS website, there's a really we talk about Nadia's story, which is I think really inspirational. But this is sort of the broad strokes of what we found, right? So of all the trials in ALS, only half of them are doing anything with biomarkers. And like none of them are doing things with mechanism-specific biomarkers. I mean, it is sorely lacking when you remove the ones that are the genetic kinds of ALS. So this year, when we rearranged, we made a lot more changes. So we rearranged our mechanisms. We made it a requirement that you have biomarkers incorporated. We were very explicit about what they must do. We added this clinical care tier to our clinical trials where this is, again, optimizing current care, what used to be in that thing called a CDA, but we've made it more money and easier to do. For the other awards, we have made it a requirement that they absolutely, you can't get one of these if you're not doing biomarker development or have biomarkers in hand. And you might have read recently about NIH saying they're going to require people to submit data sharing plans with their proposals. We don't just make them submit them, we score them. They're part of what these grants get evaluated on. We evaluate a proposal based on whether or not and how the person is going to share their samples and share their resources. We also, so there's another Curly coming up soon in May in Pennsylvania. We're going to incorporate training about how to read a grant proposal into these Curlies, right? Because if consumers are involved in evaluating the proposals, and we need to give you guys the resources and the training to do a better job. As of today, so today, the new mechanisms were just posted, and so we need to spread the word. They literally just came out today for new funding. On our website, there's also, if you go to it, a database of all kinds of open source resources. I'll tell you what's on there right now has not been updated, but it's about to be, because I personally finished the updates 4 days ago, and they're going through the DoD's process right now of getting approved. But if you check that website next week, there's a massive list of all these kinds of resources, open science resources that can be used for ALS research. So there's still a lot to do, right? And so specifically, this is kind of my wish list. We don't understand selective vulnerability. We don't understand how all these different pathways and targets can cause this unique disease. But also, if, like, for example, someone has a C9 mutation, one family member gets FTD, one gets ALS, one gets Parkinson's and FTD. We don't understand that. This is personally the thing that I'm focusing my own thoughts on the most. And I hope I have some ideas that I hope to be able to tell you about soon. We really need to be able to correlate samples and data collected during life with these findings in the tissues. And so it's important to educate the community about the importance of this kind of thing, right? It's important for us to see what we can find in muscle samples. It's important for us to keep working towards standardizing these methods and what we're doing across all these efforts so that we're comparing the same things when we get them from different people. This is something that actually I'm expecting the CDC to be instrumental in. We've been talking about this recently, about ways that we can use their platforms and resources to help with that. We still need to be funding innovative therapy discovery. I don't want trials of medicines because a pharma company says, oh, we've got some money and we have a drug, right? The point of these funding mechanisms that we've created is that they're going to allow promising therapies to get into trials, not just the ones that happen to have financial backing. We're talking to venture capitalists. We're talking to other industry reps. By the end of the month, there will be a question and answer with Dr. Lukac from Denali about just these things, about how can we help small pharma get to venture capital? How can we help de-risk clinical trials? And how can we advertise these mechanisms both internationally and also more to small pharma so they know about them, so they know they're available. As I talked about, we're doing some things to educate people. We're also, I wanted to mention, so not just the innovative therapies, so the CDC each year funds grants, and in the past, they've always been like big grants that require lots of data. This is the first year they've added a new mechanism for small innovative trials that maybe are new ideas without a lot of data, just some idea that someone has about risk factors. And so tell the people like this is a new funding mechanism. I don't think many people know about it, and I think it's worthwhile. So the last thing I just want to mention, I know I talked too much and too long, it's a sickness and I apologize, but is this idea about figuring out ways for funding agencies to work together. And so there's something that I would love for you to take a look at that's in the cancer field. It's called the ICRP, and there's the website right there. And what this is a giant grant coding effort. So all funders in the cancer space code all the awards that they give, code their portfolio dollars and what they're spending. And so there's these tools on here where you can look up, if I'm interested in prostate cancer in left-handed dentists without tonsils, right? I can go on here and I can figure out who on the globe is funding that. And I can figure out what their investment is. And I can figure out how, as a funding agency, I can best fit into this infrastructure. And I can tell you more about this at another time. So we're working, we've actually spoken to the ICRP. They've given us all of their manuals, SOPs, data elements, processes, contracts, everything. So we don't have to reinvent the wheel. We can literally just apply what they have created and is very successful to ALS. And so this is like precision medicine for research, right? One of the things that this can do that I'm really excited about is we actually added language to the DoD announcements this year to let researchers know that there's going to be a mechanism, if they want, whereby if their grant is not funded, that we hope to be able to share their unfunded proposal with other agencies in the space. And this is the kind of thing that this kind of platform can do, right? So normally somebody writes a grant, it either gets funded or not, and it's like a year before they can write that grant again. And so instead, what we're hoping to be able to do is for proposals that just maybe missed a cutoff or maybe didn't quite fit with our portfolio needs or what our mechanisms were, this will allow other funders in ALS to have access to those and perhaps fund them. So, my acknowledgement slides are ridiculous. I never know what to do because literally the world is our lab and it's super fun. And I am immensely grateful to every single person and scientist and agency in the ALS community because I get to spend my time talking to researchers about how to figure stuff out and how we can work together to figure stuff out. And none of it would be possible without all of our patients, obviously. And the two people at the top, that's Kathleen Wilsbach and Catherine Gallo. They're my two research coordinators who actually do everything, and I just babble about it. And so I'm immensely grateful to them as well. So thank you.
Well, thank you, Dr. Ostrow. We definitely gained a lot of information. Like I said before, my name is Sarah. I'm one of the Everything ALS team members. I'm also a medical student at University of Michigan. Lost my dad in 2018 to this. But myself, along with Casey, one of our student ambassadors, are going to go through some questions that we've had in the chat. So we'll get started. Excuse me. The first question is, what currently provides you with the most hope for the future of ALS?
So, I mean, you guys are my personal Kool-Aid, right? So as far as the most hope is that every time I interact with people with ALS and caregivers, it makes me want to do a better job and work harder. And so personally, that's what drives me. As far as therapies for ALS, what's providing me with the most hope is that for the first time, I think ever, and because of advocates, we may have the funds to truly bring forward things that have the best shot of working, right? I'm tired, like I want therapies for my patients that I don't need to go and reanalyze the data 6 ways from Sunday. And, I want things coming, I want to have therapies ready for the platform trial with biomarkers in hand that really look promising. And, it's because of the advocacy that you guys have done that we're positioned. And so now what we need to do is get the message out. We need good proposals, right? We need industry sending us good proposals. We need academics sending us good proposals. And so that's what I think in a broad sense gives me the most hope. I don't know what's going to work for a therapy. And even if I told you what I think is a good idea, like don't listen to me because we've been wrong over and over and over, right? But the fact is, I think for the first time ever, we're working towards having these mechanism-specific pharmacodynamic biomarkers, and we're going to figure it out.
Hi, Dr. Ostrow. Thank you for your presentation. My first question is, why is a trial considered a failure when it works for a subset of patients?
Yeah, I mean, so the whole point is that this is the frustrating thing that drives us all crazy, right? So most therapies that are tried in ALS are tried in ALS because they originally worked in some flavor of a lab assay, right? A mouse with a particular mutation or iPS cells or whatever it might be. And it's ludicrous for us to think that they're going to work for every patient. But the trick is we have to be enrolling the right patients in the right trials. And that's why we desperately need these predictive biomarkers, right? So predictive biomarkers, meaning ones that tell you who's likely to respond to a therapy. And the only way we're going to get there is having some way to validate that these things that we see in lab models are actually happening in our patients. And that's why, you know, the biosamples and the very rigorous data are so important. But I mean, call it a failure, call it a success. Like the point is, I want to know what therapies to give to my patients that are going to work for them. And this is the roadmap to get there.
Before we say the next question, I just want to make sure that you see the chat. Lots of people are very excited about your presentation. But in your discussion, you talked about the eye muscles. And so a question came up saying, are eye muscles always spared in ALS? And what are your theories as to why?
Yeah, so it's fascinating, actually. So they're not always spared. They're involved late and they're involved in specific ways. And we've learned with some recent technology, tracking eye movements. And in fact, there's perhaps going to be a camera app that does this, that there are subtle things that we can detect. But in a very broad sense, yes, they're spared. So there's some weird things about ALS that we don't understand. So, and let me step back for a minute. I deal with other nerve and muscle diseases, right? Things like myasthenia gravis or, you know, myopathies, myositis, neuropathies. Most of those diseases preferentially involve eye muscles, you know, and so it's weird. We don't quite understand why they are spared. And it's not just the eye muscles. It's also, for example, the muscles that control voluntary control of bowel and bladder. So those muscles are also specifically relatively spared in ALS. And it gets back to this idea about selective vulnerability. So why are certain cells involved and other cells not involved? And so people have done things, for example, where they've taken the parts of the spinal cord that control the eye muscles, and they compared those to the parts of the spinal cord that control, you know, the tongue muscles, right? Because tongue is really involved in ALS. And they've said what's different, and they found some things that are different, and maybe some of those are some of the reasons. But the truth is, we just don't know the reason. I do have a personal theory about it, but and it has to do with where the nerves are traveling. But again, I have to prove it. So.
Due to the large variability in pathways for potential causes of ALS, what besides genetics are we determining how cohorts should go together?
Right. So, you know, anybody in ALS any ALS researcher who's ever written a grant to a non-ALS funding agency, the grants always come back with the same criticism. It says, how can your pathway possibly be important in ALS when it's also important in sleep apnea and liver cancer, right? And it's a valid point. And, you know, and so we don't know that answer. But as far as how to define cohorts better, what we do know is that when we analyze patient tissues, that we see distinct clusters of patients, right? So that was what I was showing by the work by Dr. Hamel. So she took all of our tissue samples, multiple different regions of the nervous system from all the patients, from all the autopsies, and had a computer do an unbiased analysis, and they fall into clusters. And so some tissues had problems with, you know, oxidative stress. Some had problems with mitochondrial trafficking. Some had problems with retroelements. And so what you'd like to be able, so in other words, we can tell after somebody dies who might have been more likely to respond to a given treatment based on the mechanism. And so now what we have to do is figure out how to tell that while somebody's alive, how to match something that we can measure on an image or in the blood or in the spinal fluid or in their muscle with what we see in those tissues, because that's the, you know, the pot of gold at the end of the rainbow. Once we can do that, we can specifically enroll the patients that have specific mechanistic problems into the trial that's targeting that mechanism.
Next question kind of is a little bit different than the presentation, but how can the ALS community and advocacy groups specifically help support current trials and discovery efforts?
Yeah, so lots of ways. Number one, first of all, you guys do an amazing job as it is. Like, I don't think there's any disease-related community, anything like ALS. Maybe I'm biased and don't know, but it's not possible. So number one is, you know, when you are doing advocacy, when you are talking to communities, when you are talking amongst support groups, to really impress upon people how important it is to participate in these biosample collection efforts, these longitudinal efforts during life. Sign up for the CDC registry, participate in CREATE, participate in, ALS TDI. I don't care. Like, we all need to be talking to each other, and whichever one it is great. But do all of that, and do all of it as much as you can possibly stand to do, because that's critical. And then, really understanding the importance of autopsy donation, because that's where we're going to figure out, can we match what we see once somebody dies with during life. And so when I told you about that study that I did in the PET ligand, we had a terrible time recruiting for that study. And that was at Hopkins, where we do a lot of trials, right? And we had a terrible time because of understanding concern on the parts of persons with ALS, that if they said yes to that trial, that they weren't going to get selected for the super awesome treatment trial of the magic juice that we have in the closet, right? Which nothing could be further from the truth. And if there's one thing that would be most helpful for moving that side of the research forward, it's really understanding the importance. And then 2 is helping us outreach to international researchers and to industry. So we're actively speaking with venture capital. We're asking them, what are the problems with when pharma comes to you with their ideas and you're like, I don't want to fund that, like, why? We know what their answers are. It's always the same. They need pharmacodynamic biomarkers. They need predictive biomarkers. But we need your help with the outreach. So when we have these new mechanisms from the DoD, when the CDC has these new risk mechanisms, it doesn't matter if people don't apply, right? It doesn't matter if we're not getting good proposals. And then we also need your help, at least on the DoD's part, in literally being involved in the review process and the programmatic planning. And I know some of you on this call already do that. And I encourage you to look on the website and talk to some of the others who've done so.
Well, thank you.
With all the different studies worldwide and data on ALS across different sources, is there a single place where this is all collected? And are there supercomputers used to analyze all of this data?
So this is the crux of the problem, right? So here's the thing. It's all these regulatory hurdles that are very different in different countries and very different even at different institutions. And so number one, as you guys know, data has to be de-identified, right? And so you get these ways that you get assigned a unique de-identified thing that follows your data around that can't tell who you are. By the way, almost every ALS patient I talk to is like, screw that, use my data for whatever you need to figure out ALS, but that's a whole separate issue. But I get it. So one of the problems we have in ALS is that there's a bunch of these different identifiers. And what we need is a program, whether it's at the CDC or somewhere else, to administer a global unique identifier that truly is global, that's something that you sign up for when you get diagnosed, at your doctor's office, that follows you through clinical trials, follows you participate in like a biosample effort, follows you if, once you die and you happen to get an autopsy, so that we can easily link those samples. The other thing is that when you're involved, as you guys know, in a clinical protocol, you sign all these consents, right? After death, it's actually slightly different, but there's still a bunch of regulatory issues that have to do with how things can be shared. And so we need to educate all of these efforts about, so number one, having broad consents. If you consent to a program that collects your blood for ALS research, well, guess what? Actually, those samples can't be used for FTD research or for, PLS research. And so it's very important that consents have broad language. But the way to do what you just asked, the way to have a central connecting thing, is very straightforward. You have a checklist, a regulatory checklist, that every time you are incorporating a new resource, whether it's a analysis tool or a biorepository or a data collection, you check off, like, who can it be used for, right? What kind of research can and who can do the research? And then once you flag those things, it's simply a matter of people writing what are called APIs, which is ways that different platforms talk to each other. Because what we don't want a one-size-fits-all giant, enormous repository. What we want is to be able to connect all these various resources around the world in a regulatory compliant way that harnesses everybody's individual expertise, right? So we need to have the different analysis tools and the different data sets all be able to be easily talk to each other. And that's a regulatory issue, not a technology issue. And the regulatory issue is a simple checklist and then writing APIs. And I could talk about that for an hour, but I'll stop anyway. We have for what it's worth, we've spelled out a roadmap to do that. And I'm happy to share it with you guys and what it would take and what the action items are and the calendar items. It's pretty straightforward. It's supply chain work. It's like supply chain if you're doing anything. So.
Absolutely. Can you speak a bit about the process of validation for these biomarkers and what specifically in ALS would a new potential biomarker have to be compared to?
So, somebody mentioned a few moments ago about like what calling a trial a failure or not, right? So at the end of the day, what matters for a trial approval is some measurement that is accepted by the FDA as being meaningful, right? And so there's different ways that can happen. There's actually something called a context of use statement, which is a way that a biomarker gets formally approved in a disease. You don't actually need a context of use statement to use biomarkers in early phase trials at all. I don't even think you need them for phase 3, but you may. Somebody can check me on that. But my hope is that by doing a better job of segregating the right people into the right treatments, that I want it to be obvious, right? I truly believe that if we are choosing the right therapies for the right subgroups of patients, that if they work, we're going to see it. But as far as what's involved with validating a biomarker, it's the kind of thing, like I invite you to look at that paper that we did on neurofilament. So it's making, how does it behave in different assays in different commercial labs? What's the reproducibility, the interassay variability, all that kind of stuff. And all of that kind of data you then bring to the FDA and you say, here, look, we think this makes sense. There's actually, if you send me an email, there's some good resources about it. It's a pretty complicated process, but I guess what I would say is like the low-hanging fruit, forget about the details of the formality. The low-hanging fruit is that when we have therapies coming forward to early phase clinical trials, we should not accept that if they don't have biomarkers in hand, right? And biomarkers in hand that have peer-reviewed literature that say that they work. We just shouldn't accept it. It shouldn't be a thing because we're going to do these trials, and at the end of them, we're not going to know why they didn't work.
Speaking my language.
The next question is a multi-point question. So, can tissue donations be made while a person with ALS is still alive? Where can patients of ALS sign up to donate post-mortem tissues? And can you be both an organ donor and also an autopsy donor?
Okay. So first of all, if I take your brain while you're still alive, I go to jail. So that's just to be clear, right? I can't do that. However, that's why I'm very excited about muscle biopsy, because I do think that that's a potential tissue that can be sampled during life. Now, post-mortem autopsy donation is different as far as, so when you sign a consent for a clinical trial, so after you die, you're not considered a human subject. I didn't make these terms, don't yell at me, but so you're not, by HIPAA, you're not a human subject. So it's not human subjects research to do things on autopsy tissue. And in this country, there's a rule that says, so even if you sign a consent during life that says, I want to donate my autopsy, it's actually up to your next of kin after you die to say yes or no. And so autopsy tissue donation depends on that next of kin. And that can be a challenge. Now, in order to make these tissues valuable, autopsy has to be done within a certain amount of time and in a certain harmonized way. And these arrangements have to be made ahead of time, because at the time of death is not the time to be figuring this stuff out. And you really got to have it set up with the funeral home and the transportation and the different forms of ID that have to come with you when you come and all this kind of stuff. And so we have standardized those SOPs, and we're talking with the CDC right now, because what we'd like to be able to do is have a system where any academic center in the country that can conform to our SOPs, which are pretty straightforward, that patients seen at any of those centers could get an autopsy there, and the tissues and the data would be collected according to this, standardized data that we've created and sent to us and then used openly, and we can reimburse these institutions each time this happens. So right now, if you contact me, or there's actually, what's even better, once the DoD update goes up, one of the pages on that resource list on the DoD website is all the different post-mortem donation efforts that we're aware of. And so you can go there and look. It's just something, if you talk to your neurologist, they can give you information about it. Now, your question about organ donation, it actually is easier for us when people are organ donors, because when you become an organ donor, most states have like an anatomy gifts registry, right? And they supply cadavers to medical schools and they supply organ donations to different places. And when that happens, it makes the regulatory hurdles easier for us. And actually those organ donation agencies will handle the transportation and will take care of all the rest of that. And so, so yes, you can do both. It also doing both, if you're somebody who doesn't want to pay for funeral costs, the organ donation services do that. And so it's something we do arrange with a lot of patients. Did I answer all those questions? Did I miss one?
You got them.
Okay.
The next question, it says the NIH is in the process of developing a strategic plan for ALS. What are your recommendations for the 2 to 3 things that are important action items for the committee to focus on? I'll give you a little more context.
Yeah,
so
I know all about it, and I've given a fair amount of feedback already, and that's why I'm sort of hesitant to answer. I think that I don't want to be an expert voice in that answer. I want this to be crowdsourced, and it has been. I think that you know, the stuff that we talked about today are the So for example, the NIH has clinical trial funding mechanisms that have been in place for quite some time, and nobody uses them. And nobody uses them because the bar is too high, and nobody knows about them, truly. And so, you know, we need to, it's a horrible term, but people call it the valley of death, right? To bridge this thing from the research lab to the clinic. And the only way that we're going to do that is if we get better biomarkers. And I don't necessarily mean endpoints, right? People talk about, oh, we need something besides the ALS-FRS. We really need predictive and pharmacodynamic biomarkers. And if we can figure out who to test which drugs on, and if we can figure out when the drugs work, those are the critical parts that are missing. Now, the other thing that the NIH, you guys remember a couple of years ago, they had this, I don't remember what it's called, but there was a new ALS granting mechanism that was for these collaborative, innovative teams. I don't remember what it's called, but in any case, they often do these things where they solicit, you know, topics, right? But what's not always made clear is that that doesn't mean that specific money has always been earmarked for that. So there are certain diseases that the NIH has truly earmarked money for. So dementia, right? So there's this dementia research fund, you know, but in most cases, ALS research, like they say, oh, we'd like to see a proposal in this, we'd like to see a proposal in this. But the pot of money is the pot of money, and it's the grant coding after the fact that decides if it's an ALS proposal or a, you know, FTD proposal or something else. So I think that, you know, in general, the NIH doesn't do much disease-specific stuff, and I don't know if I would recommend it. But what we need is truly to be able to tell who to do what therapies. And you know, and one other thing is that they really have the ability to do. Like so they recently put out this thing about data. So if you write an NIH grant right now, you have to include a data sharing plan, right? But let me tell you what, a lot of agencies have done that for a long time. And researchers, it's not that they game the system, but they know how to hit the checkbox. And the checkbox is, well, we're going to share the data at meetings and, you know, scientific presentations and publications and blah, blah, blah. So no, it is important that the data sharing plans be scored. And there's no excuse. There's simply no reason not to. IP is important to protect. It's not, we're not asking like a pharma company to share their, you know, their magical drug and have somebody else steal it. But you can evaluate a data sharing plan in a proposal just like every other component of the proposal. And it's, in my mind, really, really a failing of this NIH plan right now that they're not making that required.
Thank you.
Do you know of any studies looking at tissue samples and slow progressors? Do you believe that there would be any benefit to looking at slow progressors' tissue longevity?
So every autopsy that we collect, we have detailed clinical data on, right? And we can look, we have all that. We have slow progressors, fast progressors, we have upper motor neuron, lower motor neuron, you know, every clinical component. And so when somebody comes to me and they say, I have a target that we've discovered in a lab in cells, and we want to, you know, validate it in human tissue, and we think that this target is important in slow progressors and only important in the spinal cord, and, you know, this is the kind of cell we think it's important in. And so what we then do is we actually make a sample set for them where we send a blinded test sample set, what's called like a black-white set, where we've got slow progressors and we've got fast progressors, and we've got, you know, the different regions of tissue. And so that's exactly what we do. So all the time, if somebody has a new ALS idea, their idea is, hey, I think my thing corresponds to TDP43, right? And so we send them tissues that we know either have a lot of TDP43 or a little TDP43, or happen in patients who have limb involvement or bulbar involvement or fast or slow. And this is why it's so important that when the tissues are donated, we have these standardized data sets with them. And you also have to think about when the data was collected, right? Because if somebody was upper motor neuron early in their disease, or that changed, or maybe their EMG showed something different. And so we spend a lot of time thinking about which clinical data elements matter to each project. But yes, absolutely. We want slow progressors, fast progressors. We want every single flavor of ALS, and we want the corresponding data, because that's how we figure out what's different about people.
Our next few questions come from the emails that people have sent in prior. The first one says, what is your recommendations about Radicava?
So just broadly recommendations about it.
That was the question.
So first of all, I was on the medical advisory board when it was approved, so I don't necessarily feel that I'm a good person to, I'm not on it, but I was the year it was approved. But, you know, what I wanted to talk about today isn't the existing treatments that help us a little bit. What I wanted to talk about today was how are we going to find treatments that really freaking matter. And so whenever a patient asks me about any treatment, off-label, on-label, whatever it is, what I do is I present the data to them, and I present the risks and the benefits and what is the actual data. And I inform them, because I work for you, right? And so it's up to you to make that decision. And, you know, my job is to answer questions each time a patient asks me about any therapy. And so I'm also part of ALS Untangled, right? And so that's another thing that is just really, really important, that we're giving you guys data on off-label things and things that aren't improved. And, you know, that's my job. My job isn't to tell somebody, yes, no, do this. My job is to make sure you understand what it's based on, the risks and the benefits, and help you make a decision. So how's that for a cop-out? I don't mean to be a cop-out.
No, you're totally fine.
The fact is, the title of my talk is,
yes,
we need to do better, and damn it, we need to do better, okay?
Yes.
And I recommend the existing medicines to my patients because I think they do work and they do slow the disease down, but that's not what I want to talk about today. What I want to talk about today is we freaking need to do better.
Yes, 100% agree.
Next question is, what are your thoughts about Lyme disease and ALS?
So I think that understanding the environmental exposures and ALS are very important, whether they're infections or toxins or whatever it is. I can tell you that, you know, so Lyme disease can cause a specific thing that sort of looks like a particular flavor of ALS. And what I would encourage you to do is go read the ALS Untangled review called When ALS is Lyme, because we talk about it in that, and we talk about what it looks like. The thing about ALS that's hard to kind of get, so I'm a neurologist who treats inpatients at Hopkins, right? And like, we're a tertiary referral, so we get patients who've been seen 14 other places and can't figure out what's wrong with them. And, you know, Lyme disease is actually easy to diagnose. And the people who say otherwise, it's not really evidence-based. And it's also easy to treat. And I'll tell you what the treatment is that works for Lyme disease? It's ceftriaxone. And so if there was a big correlation between Lyme and ALS, do you think that ceftriaxone trial would have failed? I mean, it's hard to reconcile that. There are patients who get Lyme disease who get something that infects their nerves. When it is affecting their nerves, we can tell by, you know, findings in the spinal fluid, findings otherwise. But importantly, they also have the sort of standard Lyme, right? Like they get the joint aches and they get the myalgias and the, you know, all the rest of the stuff that goes along with having Lyme. So read the ALS Untangled review, but it's certainly not a broad cause.
Thank you. We only have a few more. Can you please discuss your thoughts on the importance of nutrition and gut health in ALS?
Yeah. So that's a fantastic question. So first of all, right now, maintaining your weight slows the disease down better than any drug I can give you. You know how we found that out originally? I'm using the big we because it wasn't me. But you know, if you're in a clinical trial and you get a drug, one of three things can happen. You can either get worse, get better, or stay the same, right? And so some of these drugs that worked in mice or in lab assays, when they were, as you know, when they were taken to people, they actually sped up the disease. And when people went and looked back and said, what happened? Why'd they speed up the disease? It was, oh shit, that's a drug that has a side effect that it causes weight loss. And that was the first hint of a signal that, you know, that that losing weight speeds up ALS and that maintaining your weight slows it down. And so it is absolutely important. You know, I'm a big advocate for early PEG tubes, for doing, I hate it. And that's because I don't want my patients to be freaking out. Oh my God, I've got to eat all the time. I'm so, you know, I don't want you guys to, like, it shouldn't be something that affects your daily life, but anything that can be done to maintain weight is critical in this disease. And then as far as the gut health thing, I'm not sure what specifically we're talking about, but, you know, there's all this interesting new stuff about the microbiome in ALS. And so so again, there's an ALS Untangled. I was actually the primary author on that one, looking at fecal transplants and restoring the gut microbiome. And, you know, this is a hot topic of research. NIH has a whole separate division looking at this in different diseases. And there's something there. I don't know what the answer is yet. You know, my colleagues who do that research, I like to tell them all the time that their research is complete shit, because it is. They're looking at anyway, bad joke. But the jury's still out. But I can tell you, you know, it's not a sexy thing, but maintaining your weight slows the disease down better than any drug that I can give you.
Is there a way to get trials to rural areas to give these people a chance to partake?
So forget just trials. How about multidisciplinary care, right? It's really hard. And so Terry Hyman Patterson at Temple, some of you may know her, in the ALS Hope Foundation, is really doing a lot of work on figuring out how we can get, so right now, all the big centers, Hopkins, whatever, you know, we've all got these foundations associated with us, right? It's like alphabet soup, you know, Packard Center, or ALS Finding a Cure, or ALS Hope Foundation, whatever. And that's because insurance doesn't actually pay for the standard of care, the established standard of care in ALS. And so these foundation funds are needed to, in fact, just pay for the standard of care. And that is really terrible if you don't live near one of those centers and you're out somewhere rural. And so, you know, Terry is doing some studies right now on what is the difference in quality of life and the actual cost of care if you live near a center or if you don't live near a center. And the reason for doing that is to then be able to go, number one, to the insurance companies and say, you have to reimburse for multidisciplinary care in ALS. Number two, to go to the government and say, you need a freaking billing code for ALS multidisciplinary care. Right? There needs to be at least, as a low-hanging fruit, a billing code for these things that are standard of care that are part of taking care of an ALS patient. Now, carrying that forward to clinical trials, it's obviously even worse. And, you know, all of the neurologists, like, I'll tell you, like, we would love to have our patients try everything, but it's the amount of regulatory work and the hurdles, and especially if you're not associated with a center that's got, you know, trial coordinators and people to organize those trials, it's really, really, really challenging. My hope is that we get enough enrollment in these trials that, in fact, we have treatments, and so then it's less of an issue, right? But, you know, but I don't have an answer beyond that. I mean, I think, I can tell you, Terry Hyman Patterson, doctor, is planning to send out a survey, and the survey is gathering that information to help argue to the insurance companies. And she's going to need your bullhorn to help that. And she's going to need your bullhorn in particular to help get those surveys out to people who aren't affiliated with these large academic centers or neo centers.
Dr. Hyman Patterson was actually here, I think, about a month ago. And so
yeah, she gave a phenomenal talk about respiratory use, right?
Yeah, she was
so
great.
I'll tell you, she actually, when I said that we have a disease, so she's one of the people that's on the programmatic panel for the DoD. One of my first things that I did when I became chair was added her, actually. And so that she was the inspiration for the award mechanism that we have that's looking at how to optimize aspects of current clinical care, like respiratory care, right? Because that's not something that's easy to fund, and we don't know. We don't literally know what are the optimal settings and how do we decide and how do we evaluate patients. And so that she is the inspiration behind that award mechanism.
Yes, she is wonderful. This is our final question. I know we've taken quite a bit of your time, so we do appreciate it. But what are your thoughts on the research coming out from Duke University by Dr. Bedlack in regards to ALS reversals?
So again, what are your thoughts is the kind of thing I could talk over a beer for 2 hours. Here's my thoughts. I love Dr. Bedlack. He, along with Dr. Hyman Patterson, are two of my heroes, and I strive to emulate them every day. It's fascinating. When I look at other diseases, for example, HIV, right? So HIV, once upon a time, they said, you know, let's look at what they called long-term non-progressors, people who had HIV and yet never got AIDS. And let's see if we can figure out what's different about those people. And then let's see if we can study it. So I think we need to there are definitely patients who meet the criteria for ALS and their disease stops or it reverses. And we need to learn from them. You know, there's people, for example, in the traumatic brain injury field. So there's boxers, right, who get punched in the head their whole life and never get traumatic encephalopathy. So what's different about them? You know, why doesn't a woodpecker get, you know, brain trauma from hammering on trees? Studying the ALS reversals to me is that same thing, and it's critically important. And I love that he's doing it, and he's the right person to do it, and I can't wait to see what comes of it. By the way, a similar parallel in the mice. So we learned early on in the mice experiments that if you just breed these mice with a different, the control mice, a different what's called a genetic background, right? That they live longer. And it was screwing up a lot of experiments, because if you cross an experiment and you saw them live longer, you're like, oh, look, my therapy worked. When in fact, no, it was just that the strain was a little bit different. And Dr. Patterson is somebody who looked at that and said, whoa, everybody's throwing that away and saying that that's an artifact and screwing up experiments, but why does that happen? You know, what's different about this genetic background? What are the modifiers that's making it so that ALS is faster or slower? So these are the kind of things we can learn.
Wonderful. Well, like I said, we are so grateful for your time and all this information. I hope you're looking at the chat, because people are just thrilled with this presentation and all the information and the way that it was presented. So thank you very much. As for everybody.
Real quick.
Yeah,
so
absolutely.
First of all, don't thank me, because I love to talk to you guys, and I'm happy to talk anytime, and anyone can reach out to me by email, whatever. I'm happy to talk. You know, I want to figure this out, and I work for you. So please, you know, always happy to talk.
Wonderful. We did have one request for potentially getting to some of the slides. I think there was some interest in being able to reread some of the slides more than just on the recording. For everyone on the call, as you guys know, this is our time for open forum. We turn off the recording and let you guys kind of discuss what was said, what was talked about, and what's important to you. So again, thank you. And we will absolutely be seeing you in the future, and hopefully for big and better things the next time around too.